50S is the largest subunit of the 70S ribosome of prokaryotes. It is the site of inhibition for antibiotics such as macrolides, chloramphenicol, clindamycin, and the pleuromutilins. It includes the 5S ribosomal RNA and 23S ribosomal RNA. Structure: 50S, roughly equivalent to the 60S ribosomal subunit in eukaryotic cells, is the largest subunit of the 70S ribosome of prokaryotes. The 50S subunit is primarily composed of proteins but also contains single-stranded RNA known as ribosomal RNA. A RNA forms secondary and tertiary structures to maintain the structure and carry out the catalytic functions of the ribosome. X-ray crystallography has yielded electron density maps allowing the structure of the 50S in haloarcular maris mortuae to be determined to 2.4A resolution and of the 50S in Deinococcus radiodurans to 3.3A. The large ribosomal subunit is approximately twice as massive as the small ribosomal subunit. The model of HM50S, determined in 2000 by Nenad Ban and colleagues in the laboratory of Thomas Stites and the laboratory of Peter Moore, includes 2,711 of the 2,923 nucleotides of 23S RNA, all 122 nucleotides of its 5S RNA, and structure of 27 of its 31 proteins. Ribosomal RNA. The secondary structure of 23S is divided into 6 large domains, within which domain V is most important in its peptidal transferase activity. Each domain contains normal secondary structure and is also highly symmetric in tertiary structure. Proteins intervene between the helices. At tertiary structure level, the large subunit RNA is a single gigantic domain, while the small subunit contains 3 structural domains. This difference reflects the lesser flexibility of the large subunit required by its function. 50S includes the activity that catalyzes peptide bond formation, prevents premature polypeptide hydrolysis, provides a binding site for the G protein factors, and helps protein folding after synthesis, promotes the peptidal transfer reaction and prevents peptidal hydrolysis. An induced fit mechanism has been revealed for how 50S catalyzes the peptidal transfer reaction and prevents peptidal hydrolysis. The amino group of an aminoacyl tRNA attacks the carbon of a carbonyl group of a peptidal tRNA and finally yields a peptide extended by one amino acid esterified to the A site tRNA bound to the ribosomal A site and a deacylated tRNA in the P site. When the A site is unoccupied, nucleotide U2620, A2486 and C2106 sandwich the carbonyl group in the middle, forcing it into an orientation facing the A site. This orientation prevents any nucleophilic attack from the A site because the optimal attacking angle is 105 degrees from the plane of the ester group. When a tRNA with a complete CCA sequence at its acceptor stem is bound to the A site, C74 of the tRNA stacking with U2590 induces a conformational change in the ribosome, resulting in movement of U2541, U2620 through G2618. The displacement of bases allows the ester group to adopt the new conformation accessible to nucleophilic attack from the A site. The N3 of A2486 is closest to the peptide bond being synthesized and may function as a general base to facilitate the nucleophilic attack by the amino group of the aminoacyl tRNA. The pKa of A2486 is about 5 units higher in order to hydrogen bond with the amino group, thus increasing its nucleophilicity. The elevation of pKa is achieved through a charge relay mechanism. A2486 interacts with G2482, which hydrogen bonds with the buried phosphate of A2486. This buried phosphate can stabilize the normally rare amino tautomers of both bases, resulting in an increase in the negative charge density on N3, helps protein formation. After initiation, elongation, and termination, there is a fourth step of the disassembly of the post-termination complex of ribosome, mRNA, and tRNA, which is a prerequisite for the next round of protein synthesis. The large ribosomal subunit has a role in protein folding both in vitro and in vivo. The large ribosomal subunit provides a hydrophobic surface for the hydrophobic elapse step of protein folding. The newly synthesized protein needs full access to the large subunit to fold. This process may take a period of time.